I'm really here just to introduce Mathilde, so I'll do that briefly, and then I'll let Mathilde give her presentation. But my name is Mike, and I'm a lecturer in the department. Mathilde did her undergrad degree in biochemical engineering and had the dubious fortune of doing a final year project with me. This means obviously that I knew that she was good at engineering, but also that she has the ability to generate a lot of good scientific data. And she has the equally important skill of being able to visually present the data in innovative ways, which I think really helped her in her career. Because of both these things, I tried to keep her at UCL to do postgrad research, but she knew exactly what she wanted to do with her career. So she went off, left us, and went to work in pharma. And then while she was working again with me on the planning of a kind of new food-based education offering that we're planning to give, she transitioned into cellular agriculture. So this is going to be good practice for her. She's going to have to teach on our course. But it's also an absolute pleasure to have her come back and speak to you about the discipline. Over to you, Mathilde.
Hi, everyone. Thanks, Mike. So as you said, I joined the degree in 2015, and I'm back to tell you a bit about my time there, but also Hoxton Farms, where I'm currently working. So, oh, sorry. Where does not work? Okay, perfect. Yes. So Hoxton Farms is the company where I'm currently working, and it's a startup based in London. So today I'll go a bit through an introduction to cellular agriculture and what does it mean, why we need it, and then how exactly we do cellular agriculture here at Hoxton Farms, and then what biochemical engineering is in practice and how do we get there. And we can do questions Q&A after. Please let me know if you can hear me or if there's too much background noise, and I'll try to fix it. So I'm now a bioprocess engineer. I did my biochemical engineering degree, so both the undergraduate and the master year in the biochem eng department at UCL. And since I've pursued my career in bioprocess engineering. So I started in 2015 was the undergraduate course at UCL, the BEng. In 2017, I had a scholarship for a summer project with the department doing sugar beet based bioethanol. And then the next year, I had the project with Mike that you mentioned, the final year project where I worked with Sartorius. It's a lab and bioprocessing equipment company. And then I graduated from the master's in 2019. So I had a real graduation just before COVID. And then I joined UCB, which is a pharma company where I was an associate scientist working on process development and process optimization for a Fab antibody fragment, which was used to help with autoimmune diseases. And recently, I've joined Hoxton Farms as a bioprocess engineer, and I'm working on the process creation and development to develop a cellular agriculture product. So what is cellular agriculture? Cellular agriculture comes as a response to one of the largest sustainability issue, which is the meat industry. So it's a $1.2 trillion industry, but it has terrible consequences on the environment, on health, both of humans and animals. So the main problems are the greenhouse gas emissions that are produced because of the feed that needs to be transported to the animals, but also their processing, the storage of meat in cold rooms. There's also land and water pollution and usage, because we need a very large area to breed all of the livestock that we're consuming. And with their dejection, they also tend to pollute the soil and the water and spread those pollutants. The animals are also sadly given loads of antibiotics in industrial agriculture. We saw the effect that intensive agriculture can have on public health, and they're trying to reduce that by giving antibiotics to the animals, but we saw that it doesn't always work. And then, exactly, so antibiotics and human pathogens. So how exactly are we offering to solve these solutions? There's out in the world, there's different technical and sustainable solutions. So you find a lot of plant-based alternatives. So it can be, Beyond Meat, Impossible Burgers, even corn with mushrooms. So plenty of different solutions. Usually you can use beetroot for the coloring and a lot of peas and soy for the texture. And then we have precision fermentation. So precision fermentation is focusing on one component that can bring the taste. So for instance, Impossible Food has identified the taste of blood and is expressing that in yeast. And then we have finally the cultivated meat or fat, which is cell-based, and the product is the cells. So if we go a bit deeper in how these types of cellular agriculture work, if we start with precision fermentation, the important difference with precision fermentation and cultivated cells is that in precision fermentation, you're using the cells and you ask them to produce the protein of interest. So using genetic engineering, we can insert genes in the bacteria or yeast, and then they will produce the protein of interest in the same way our cells produce antibodies for our body and so on. And with cultivated meat, we will take some cells from the animal, so a very small portion that won't kill the animal. We're isolating the cells of interest, and then we're growing them and replicating them until we have enough to make a full piece of meat or fat. Precision fermentation is currently used a lot as an addition to plant-based options. So for instance, Impossible Food is adding that blood taste protein, hemoglobin, to their plant-based patties. But one of the big problems for plant-based solution and hybrid solution is that the animal fat is perfect because it's delicious, it's versatile, it has a function because it gives this texture, juiciness, and it cooks nicely in the pan. So how can they reproduce that? For now, unfortunately, they're using only plant oils, and plant oils are poor in taste because you can use vegetable oils, or coconut oil is very popular, but coconut oil has a very strong taste that is hard to remove from the product. So usually they have to mask it with a lot of spices, and you're going further and further from the taste of real meat. And also the plant oils usually are more liquid, and they result in a greasy product more than a juicy and a little bit roasted product. And most of the time they're unsustainable because one of the common one is palm oil, and we've seen the impact on deforestation and also intensive agriculture again. So how do we fix this? That's the mission of Hoxton Farms. And the idea is that we grow real animal fat without the animals. So it's a cultivated fat company. And the approach is that we're using the power of cell biology and our understanding, and also mathematical power, to find a solution to create more sustainable meats. This double approach comes from both our co-founders, so Max and Ed. Max is a cell biologist, and Ed is a mathematician with computational expertise. And so how does the process exactly work? If we start from the beginning, there's a livestock, so it can be cow, pigs. We take some cells in the fat, and then we isolate the specific type of cells that we want, so stem cells, and then we put them in reactors at larger scale, and we replicate them, we grow them, and then we turn them into fatty cells. Then we harvest them, put them in bottle them, and then we work with customers, would be plant-based companies. So for instance, if a company wants to add real animal fat to their sausage, they would come to us, we work on the right fat, they add it to the sausage, and then it finally gets in the supermarkets for you to buy it. So the cells we're using are stem cells. Stem cells are a particular type of cell. Everyone has them in their body, and they're purpose is that they can turn into any type of cell in your body. So, and they will understand what they need to become based on where they are and what's around them. So if everything is very hard again around them, they'll know that they need to turn into bone cells. If things are more stringy and tight, they'll know that they need to turn into muscle. If it's smoother, they can turn into fat or skin. And so that's why they're interesting. Stem cells have this power to replicate and multiply as long as they're not differentiated. So as long as they haven't choose their pathway and what they will become. So they live in two phases, what we call the expansion, where we have to preserve their potential, and then the differentiation when we start changing their environment and asking them to turn into fat cells. This is done using different types of media and growth factors. The second thing about the stem cell is that they're adherent, which means that they need to grow on something. You'll see that in production, there's different type of cells for different products, and they don't all need to grow on a surface as stem cells do. So one of the big challenges for the industry is to understand what scaffold we can create. So they can be little beads, porous beads that the cells grow on. They can be hydrogel that are molded into specific shapes, and plenty other materials. There's even some people are also working on using leaf from plants because they have a system of distribution of nutrients. So there's a lot of research ongoing on scaffolds. And the last thing is the media development, which is really interesting because we need at least two types of media, one for the expansion culture and one for the differentiation stage. Because through the media, they will understand which stage they need to be in. This is one of the big challenges for the industry because traditionally and unfortunately, stem cells media contain serum, which is derived from animals and from cows. So one of the big challenges is to find alternatives, whether they're recombinant, whether it's through cell engineering, to get to go over the use of serum. So where does the math and the computational aspect come in? The idea is that we can use the computational power at different stages. So whether it's just building app by software engineers building apps for us to use in the lab so we can make things faster, but also how we store data, how we analyze data, because we're collecting images, we're collecting genomic data, and having the computational team helps us analyze and make the most of the data, but also helps us develop new experiments and new assays that work with these computational tools. So our team is composed of synthetic and cell biologists, but also tissue and bioprocess engineers. So the bioprocess engineers will work more on the process development aspect, whereas the tissue engineer will work on the scaffold we were mentioning and understanding what's the best scaffold and type for the cells. And the computational scientist team, where you have there's some people that come from computational background that have no experience in with cellular agriculture before, some people that worked on cell modeling and so on. But what does a biochemical engineering do in practice? So the purpose of bioprocess engineer is to harness the power of cell biology sustainably at industrial scale. So harnessing the power of cell biology, as this means understanding the cells you're working with, understanding their biology, understanding if your product is the cell. So do you need to preserve the cell, or do you need to actually work with the cell so it would produce your product? And then there's all the purification steps where you need to get rid of the cell and preserve the product, which is commonly done in the pharmaceutical industry, especially for pharmaceutical products. So the difference between pharma and startups, especially in cellular agriculture, for process engineer, you'd say that in pharma, the product is very different because it's high value products. You won't pay the same thing for a dose of a medication that can help you get better and a steak in a supermarket. It's very long validation processes, so it's it's a different time scale than with cellular agriculture, and there's a lot more precision and processes that are already existing. It's also exciting because you can actually optimize processes that are already existing, and you can see the products already out there, helping millions of people living a better life. It's also larger companies, larger teams, which is very interesting for learning, and also how to interact with different teams, because you can have a team that is specialized in doing assays, a team that is specialized in one type of reactor, and so on. And it's also a good learning opportunity because you have multiple products and cell types in the same company, and they have processes that are already in places that have spent years developing and optimizing, and so you get a chance to learn how to work at this industrial standards, and that's something you can take with you to smaller companies or to your next role. And then in the cellular agriculture, so more at a startup scale, it's a smaller team, so you get more ownership on your product, you get to develop your own experiment, but it also means that you need to do different tasks, tasks that you've never done before, and everyone participates. So it's more learning by doing, which means also failing sometimes and making mistakes, but everyone does the same. So it's a very welcoming environment to learn. Compared to pharma, you will develop new processes. So everything is new. The company each month, a company gets to the next stage of development and closer to an industrial level. For now, the largest one are mostly large pilot plants that can supply maybe for one city or a few restaurants, but we're not at the industrial scale that we can see in pharma, which is a bit daunting because if you try a new process, there's no one has done it before, so you might as well you take the risk of failing more, and your decision feel have more impact on the industry. And this means that you're also creating the future of tech, and that the regulation is non-existent, which is both exciting, but also puts a lot of pressure, because if you start making decisions that won't be supported by regulation, you might have to change the way you do things. But it's also very interesting to have the opportunity to create a new aspect of our world on how we feed our population and how all of this is regulated. And finally, you're working towards an accessible and sustainable product, which is less often the case in the pharma industry, because the priority is the efficiency and the safety of the medication. So as you see, there's different skills and many technical challenges as a bioprocess engineer, whether it's upstream growing the cells or down using equipment for purifying your product or scaling up novel technologies. There's also lots of mathematical modeling or more of a biology-based position. And you can also go into pharmaceutical advisory, how to develop medication, how to increase the production. So there are as many engineers as there are skills, and starting as a bioprocess engineer doesn't limit you to a certain position because it's a very wide field. So how did I end up in that specific part of bioprocessing? I would say is through the degree and through different projects where I started learning what I liked. So everything came very naturally. So I think in my first or second year, there was a coursework that we did as a team that we really enjoyed, which was designing a process to produce bioethanol from the waste of the sugar industry. And so we had to design the process and run the life cycle analysis on it. So life cycle analysis is understanding everything that comes into your process and their impact on biodiversity, air pollution, energy consumption. And this way you can describe a process understand the carbon footprint and understand how to improve it. So this was a very interesting project, and our teacher at the time, Dr. Brenda Parker, really pushed us to go and present at the conference. So we kept working on it outside classes, and we got accepted at the conference, and it was really exciting to show our work. And following that, we wanted to get experimental data for our life cycle analysis. So I went and pestered Mike to let me run a reactor for this. And luckily, they supported me into getting a summer scholarship from the EAPSRC. So the idea is that you get paid for a month or two to conduct a scientific project in the department during the summer. So it's a great opportunity to spend more time on a subject that you liked, but also work with PhD teachers and really starting to get your hands on equipment as well. And then during my master's year, Mike supported me to get a project from Sartorius. So Sartorius is an equipment company, and they can provide bioreactors, but also equipment from the lab at very small scale. And they developed this new filtration technology to get cells and separate them from the media. And it helped a lot with one type of cells, so CHO cells, which are the most common cells in the pharma industry for mammalian cells. And they wanted to know if they could use it with algae, with yeast, with bacteria. So my project was learning how to grow all of these different types of cells and see if the technology could work. So it was amazing to have this opportunity to work both with a company, and so producing results and reporting to a company, but doing that in the lab with UCL, with people having time to teach me how to grow these cells and help me using different types of reactors in the pilot plant, which is very rare at undergrad and master's level. So this helped me get a job in pharma after I graduated, because I had worked with different types of reactors. So I joined UCB. It was a microbial process development team. And we were working on optimizing one culture parameter to increase the yield of DAPI, which is a product that people with autoimmune disease use to reduce their symptoms. And at UCB, I was also working with another associate scientist who was actually doing a year in industry from the department. So there's always connections and different opportunities to gain professional experience or lab experience. And that's finally how I got my job at Hoxton Farms, because Hoxton Farms was already in contact with UCL. And so all of this happened pretty naturally. This is one example of how you can follow a path and find the opportunities available in UCL, but I've also friends who've helped on mathematical projects or started PhD following a project that they liked with their teachers. And the department is way more than just a degree and the class time, and it's just about reaching out and going for these opportunities. So yeah, I had an enriching time at UCL. There's a wide range of topics and passionate scientists that just love to talk about their science and their project and are ready to give you time and find projects for you. There's a lot of support and opportunities. You can go to conferences, summer projects, but there's also iGEM, which is an international competition of genetic engineering where you work as a group for the summer, and then you go, I think they went to Boston a few years ago, and then you can develop your own project and have access to the lab resource to do it. That's also another amazing thing about UCL, is that you have access to a leading research center and a pilot plant in the center of London. It might sound a bit abstract, but not that many research centers have such a big biochemical engineering research department and a pilot plant that students can use. And finally, I think the classes were really tailored for the professional life. I think every week at least I go back to my classes. I have my notebooks next to my desk, and it's very finely tuned to get you set up to do your work as a process engineer. So yes, if you have any questions, this is the time, and thank you for listening to me.
Fantastic.
Yes, so the question was, what kind of feedstock is used for cultivated meat, cultivated fat? It's going to be the traditional feedstock, in the industry that we find. So it can be cows or pigs, and it also depends on what clients or the plant-based producer would like to add to his product. And maybe in the future, we can have wilder animals as well.
I think I'm going to add to it because it's actually interesting, isn't it? Because with cultivated meat or fat, there are two types of feedstock, which is the cell line itself is a feedstock, but also what you feed the cell line will also be a feedstock. And there's a lot of research going into the other one. So the food that we give to what becomes I guess a single cell is oftentimes, or like traditionally would be animal derived, but people are looking at making methods for the feedstock that you feed to the cells to be non-animal derived. So it is further removing animals from the food chain, as it were. I've got a question for you, Mathilde, which, given that you're at your current place of work, it might be hard for you to answer, but what do you think you're going to do next?
Well, I think the advantage I can answer it in the workplace is that the company is growing a lot, and soon we might be at our next scale. So developing a pilot plant, and then after that, they're developing the manufacturing side. So that's clearly one of the wonderful aspects of working in a startup, is that you grow with the company and you're always doing different things and challenging yourself.
That's great. So, but do you think you'll stay within as a bioprocess engineer within that company, or do you think you'll move away from engineering and focus more on some of the business aspects of the company, or will you always be in the lab?
So I think in the engineering team, there's different projects, and as we grow, some of us might be more specific to one. So are you working more closer to the reactor, developing the technology, or are you working more on developing and optimizing the current production? And I think I really like the production aspect of it, and so seeing how much product we can make, what type of product we can make, working with the clients, and so on. So I'll probably stick to that aspect.
Okay, thank you.
But there's a really good question has come in from Mathilde, and I'm sure Michael has something to say about this as well. Mathilde, would you like to say what the question is and what you would respond to that?
So I think for the past 3 years, Mike has been asking me every 6 months, why am I not back in UCL doing a PhD? I don't know, maybe later, but at the moment, I love working in the industry. I think that maybe earlier in the pharma, or like a few years ago, a PhD was still very important to reach some higher level positions, but seeing they're changing a lot. And I think especially as engineer, there's a lot of learning to be done on the field as well. And some we see engineers in higher positions, some did PhDs, some didn't, but there's no right or wrong answer. If you're passionate about a subject, doing a PhD is a great opportunity to go in depth with it and become an expert in the field, and that makes you very valuable as well. So I don't think, I think I didn't want to commit for 4 years to a specific topic, and I was happy to be in a more changing environment.
Well, actually, that's actually spurred a couple more questions. But Mathilde, would you consider doing an NGD with us, or somewhere else? Or would you consider doing an NGD? Would perhaps Mike would we explain what an NGD is in relation to a PhD? Because it's been very interesting working with people who are doing NGDs.
Yeah, I guess that's also a question I've asked Mathilde many times, and I have graduated slightly to like getting Mathilde to agree to supervise one if she doesn't want to do one herself. So I guess the first thing being that traditional bioprocess industries, which I guess mostly would include pharma, there was a kind of glass ceiling that you would reach if you didn't have a PhD. That is being disrupted, I think, by the startups, as I think all startups across like all industries essentially are changing the way that people work, and changing the requirement for like levels of education in some ways, and really focusing more on skills that you learn. NHDs and PhDs are fundamentally the same in that you need to do the same thing for both, which is to produce new knowledge. But NHD is always going to be, it's going to be partnered with a company. It's going to solve an industrial problem specifically, rather than just a theoretical or blue sky or interesting problem that we would create. Is solving a problem for an industrial partner, working with that industrial partner, and you then therefore taking your solution on as part of their way of working in the future. They're slightly longer. They have, so you get 4 years instead of 3, but they're all kind of being standardized in some way now. But people within engineering do prefer NHDs because of that industrial link. It gets you, especially if you've not worked in industry at all, it gives you links to industry, which is really useful for getting a job or career afterwards, as well as letting you understand how industry works.
Yeah, and I think at the time when I finished my MN, my big plan was to go to California and join a satellite company. Keep in mind that it was 3 years ago. There were very few companies in the UK doing cellular agriculture. There's still not that many, but more and more every year. And junior position for someone with only internship or summer or master's project experience, there was not an opportunity yet in the UK, and there was no NHD position that could work with the companies, because those companies were a few months old or maybe 1 year old, and at the time they didn't have the foresight to be able to commit to a PhD student or NHD student saying, well, so we can tell you that we have one problem that we want to fix over the next 4 years, and you'll be working with us on that. I think as companies develop and get to more industrial scale and have clear long-term planning, then they'll be able to support NHD students. But my goal was to join the cellular agriculture as fast as possible. So do a bit of pharma, learned a lot there, because they're already at that scale that is not achieved by satellite companies, and then bring that experience into satellite. So at the time, the best way for me to do that was to go into the industry straight away.
Yeah, and before you move on to answer the next question, Mathilde, I say one of the problems that small startups have, apart from the one you've already identified, is that they're really focused on short-term improvements to get more funding. And a NHD naturally means you've got to give someone a problem that they can answer in 4 years. And you have to get to a certain size to be able to look that far in the future.
Yeah.
Also, just one of the things that's come up a lot with people have asked, do you need a PhD to work in the sector? Why do so many of our students go on to do PhDs? And the answer that they come back to it is that it's just such a new and exciting area that when you're doing a degree, you'll get interested in something, you'll do some work, but find you're actually doing novel research as an undergraduate. So people want to run with that because they know that what they're doing is new. It's not been explored yet. So it's not that people need a PhD, but they get so excited by the subject and find that they're working on a really exciting area. They want to go through with it. So that's something which is all worth saying from other webinars that we've come up with. So we've got 2 more questions that have come in. I don't know who wants to answer which one in which order, because there's one specifically Mathilde.
Yeah, I can start with that one. So the question is, what are the greatest challenges in scalability of culture fat at the moment? So the challenges are the same across the industry. In a way, fat is slightly less challenging, because if we look at how fat is and how we put it in meals, you see that fat is not as structured as muscle. It's way easier to put on fat than muscle. You don't have to exercise your fat. You don't have to give it structure. So the biggest challenges in scalability is how do we bring it to an accessible price? And so if we look at the cost drivers for cultivated cultivated meat, is mostly the media component. So as I was saying, media is what we use to tell the cells if they need to grow, to keep them alive, ask them to grow, ask them to turn differentiated into fat or muscle. And to do that, the part of the media that is most important to do that is growth factors. So growth factor, as Mike was saying, growth factors were originally sourced from serum, which comes from fetus of cows. And so there's a lot of companies working on resolving this specific challenge by creating recombinant growth factors, for instance, where they will engineer yeast or bacteria to produce a specific growth factors at a lower cost. Then in terms of more technical and scale challenges, we need to increase the yield from single batches. So it's understanding the type of scaffold that we can grow cells on. So in the farm industry, they can use plastic or animal derived scaffolds, and that's not something that you would want in a food product. So that would be one of the other challenges. And the third one is probably finding the right cell line and how do you immortalize them. So immortalization is the process in which you engineer your cell so that it can keep replicating. And if you don't do that, you can replicate it for a few passage. So it's the name for a replication. And then you would have to go back to the animal. The idea is to keep that capacity in your cells of replicating and replicating so you can create a large bank of cells, freeze them, and avoid going back to the animal, and do that as little as possible. So those are the three axes in the scaling up of the cells, of the cultivated fat.
Yeah, and to answer the next question, which is useful because it links really to what Mathilde just said, around the regulations in place for creating cultivated meats or fats for human consumption. It's interesting because the regulations are very national and not global. There are different regulations for what counts as being a modified organism. So if we look at that element of the process across the globe, and you do have to do some form of, and also you have to do some form of modification to make your cell line immortalized, as Mathilde has just said. How you do that is, regardless of the fact that you may end up with the exact same cell line, the method you use to immortalize will have an impact on what the cell is considered and how modified it is. And then we bring in that element that Mathilde's also talked about around growth factors. If we're not using FBS or fetal bovine serum to produce the growth factors, and we use a biotechnological method for it, that growth factor is probably going to be derived from a genetically modified organism. And then that growth factor will be taken up and is in many regions not seen the same as a non-biotechnologically derived growth factor. So there are elements of modifications, genetic modifications that we have to do to create the product. Different places have different rules on what you can and can't do. The kind of idea about not having anything left in a product sometimes removes some of that. So if you can prove that anything you've genetically modified is completely removed, you can get around things, but it's difficult to do. So there definitely are, and I think as the space grows, it will have the power to change legislation within different regions.
Thank you. So we've answered all the questions that have come through so far. I put some links to other events that are coming up. Certainly they're the one coming up next week on vaccines, and some links to the open days that we've got coming up, the undergraduate, and to a form to fill in if you want to stay in touch with us about postgraduate events. I can see that we've no more questions, and so I'm going to say thank you very much for joining us this afternoon. I really hope you enjoyed this. I hope it's been informative and interesting, and I very much enjoyed taking. Thank you for your kind words in the chat as well. Really appreciate that. So huge thank you to Mathilde. Huge thank you, Mike, and I look forward to seeing everybody at next week's final Spring into STEM session on vaccines. Thank you very much and goodbye.
Thanks, Kim. Thanks, Mike. Bye, everyone.